comparative modeling already. And essentially, comparative modeling is about, I have a protein, a query protein Q, and I find a protein in the database PDB of known structure. Since I know that proteins with similar sequences have similar structure, I assume that Q and the template in the PDB have the same structure. So the first issue is to find the template, then to align the template. So these could be the same. And there could be different steps, because finding may in fact be different than aligning. So here I put down dynamic programming. So you can, for instance, if you thought about cluster omega, with cluster omega you cannot search through 111 million just because of CPU time, or you barely can do that, certainly not for many proteins. So you would do the search with PsyBlast, but you may argue that cluster is more accurate in fact aligning templates and queries than PsyBlast. So once you have the possible candidates, you may in fact do the dynamic programming. And there are sort of more refined other methods here, like profile, which in fact HHBlitz is, advanced profile methods. And then there comes this issue of build the model. Ultimately, build the model, what's the simplest way of building the model? So you have a protein, the template in the structure in PDB for which you know the structure, and you have the target for which you want to predict the structure. So the simplest way of building model is you copy the coordinates. How do you copy the coordinates? Well, from the alignment method that you run, you have corresponding positions, right? You align every single residue in the template, in the known structure, with the one in the query protein for which you do not know the structure. And aligning means that I know what the first, second, and whatever corresponding residues there are, I know where they sit. And for those corresponding residues, I copy, so in the example of my hand, I copy for the 5 residues, I copy the 5 coordinates onto my query, right? And then comes assess the model. So assess the model comes with questions such as, well, how well do these fit? So maybe I can sort of move things, and maybe can move things around such that I sort of refine the model. So maybe there are some clashes. What are reasons for clashes? Why could it be so that my 5 residues, what could go wrong? My 5 residues, that's the query, that's my template, known 3-dimensional structure. I copy the coordinates. How could that go wrong? Can you just, there are many ways in which it could go wrong. Can you just name a couple of them? Or everyone in the room. I'm just going to go through the room and everybody is going to mention one way.
You mean why it didn't match? Or?
What could go wrong? And so again, I have a template Q, I have a structure in the PDB, and typically they would be 100 residues long, right? Here in front of you I will just do 5 because I have difficulties to show my 100 fingers. But now imagine those are 100, right? So I copy the 100 coordinates over to this one. Now my query has 100 coordinates, right? What could go wrong?
Maybe they don't have the same number of coordinates.
So number one is they have a different number of residues. So, but that, in fact, in the alignment, you have every single residue aligned to another residue somewhere else, except for you have insertions and deletions. Insertions and deletions could go wrong, but for insertions and deletions, I do not have a corresponding. So if I have an insertion, so I have this one here aligned to this one. So then, in fact, I have a deletion or insertion, whichever way I read that, right? So I will not copy that coordinate. In that case, I would copy 1, 2, 3, 4 coordinates. So what could go wrong in this copy of 4 coordinates? So then we start here because Tatjana wanted to say something, but we do. So think about something while you listen.
For example, in this example, it could be too wide.
So it could happen that, so I have maybe a bad example here. So maybe one, in fact, is a much larger residue than the other one. So in my structure, I may have something that is relatively small, and I replace it by something that is relatively big. So in the structure, I don't have space for something big. It's going to sort of displace it. That's one thing that goes wrong. Something else.
Could be a problem if they are not correctly aligned, but shifted.
Yes, so this is a major thing that go wrong. So the moment I make a mistake in my alignment, I simply copy the wrong coordinates, end. And so at this level, so I would call that a major mistake. Yes? What else, Luisa? Or Lisa? Mariana?
Well, at the end you will have different secondary structures.
So you could have, so what could be is that you believe they have the same 3D structure, but they don't. Ultimately, this leads to different secondary structure, right? Because if they have if they have different secondary structure, they at least locally have different structure. Meaning that your decision to copy the coordinates was a mistake. Yes, that could go wrong. Florian?
Yeah, when residues don't match the referring structure, like totally.
So it could be, for instance, that we have really, so in some sense this is the small versus large, so we bring in new biophysical features, and these new biophysical features such as the proline, so in particular in the context of secondary structure, this may just introduce a new kink, it may introduce something that really would locally change the structure, and then something would be slightly different here. Yes, that could be something else. I still go around, there is one person, Louise still, you still owe me one. Luna. What could go wrong? Martin. Pierre?
I don't think so.
Yeah?
To give the chance for missing spots, like
Missing spots, you mean like insertions, or missing spots what?
not structurally, like whether some residues you don't know the exact position of them, it's just a kind of blur.
Ah,
I don't know if
oh, that's an interesting one. But then you cannot copy the coordinates, right? So this is an interesting problem, and you're totally right. That could go wrong. But this sort of is, in some sense, is before what I'm starting. So it's not going wrong from copying the coordinates, but the step itself cannot happen because you don't have a coordinate. Yes, that could go wrong. So that you read of that sequence, but in the PDB, you actually for the Q don't have the structure. Or you have a diversity of states or something like that. Yes, that could go wrong. We're going to get into more details of what can go wrong before in a moment, just sort of generally. So when you look at pairs of proteins, so the query to the template in the PDB, and we're looking at the pairwise sequence identity, if it's very high, then the problem is speed of modeling. What speed of modeling actually means is if I have these clashes, so let me go back to this slide here, if you have things that ultimately come from large residues or from kings or from really different biophysical features that would induce different secondary structure, then what you try to do is you try to change the model such that you get a little bit more space. For instance, right? If it's too large. Or you try to change the angle to move it around so that the proline would actually fit into this changed, right? But this, in fact, if you change the model anywhere, this is going to have another effect. So if you make it a little bit bigger, maybe that clashes somewhere else, right? So it becomes an optimization procedure. And this optimization procedure, in fact, takes CPU time. That's why this is sort of speed of modeling. That's exactly what is meant here. Here, in this realm, it's essentially the quality of the model. That means we are getting into a realm where they have diverged so much that you have, in fact, the proline really inducing a slightly different structure. And you cannot model it because it's a different structure. It's slightly different. Maybe it still has the same binding site, maybe it sort of still does the same thing, but they're local different things. There may be different specificity. Maybe it binds something else, or there may be all kinds of other reasons, which affect the quality that you can build a model. Then if they diverge more, this query and the template, then you get to the point where in fact the mistakes that you have in the alignment, I can't remember who said that, you said that, so the mistake in the alignment become the major thing. So the more two proteins differ, the more you will make mistakes in the alignment. At two proteins for 50% identical, every alignment with 50%, there are some mistakes. That means you will read off some wrong coordinates. And the moment you read off one wrong coordinate, you sort of implicitly read off two. So you read the wrong one that you see, and you made another mistake in the one you should have seen, right? This is sort of a double mistake every single time. And then if you diverge even further, the problem really becomes the one of detecting the homology or the relation structure. That means there is a protein in PDB that has a similar structure, but you don't know which. Okay? If you knew the structure, you could find it, but then, this doesn't really help you. State-of-the-art methods. So one of the major methods in the field historically, the major method is so-called modeler from the Charlie lab at UCSF. That's Andrei Charlie, who did his PhD with Tom Blundell, went on to Martin Karplas at Harvard, became a professor at Rockefeller University, is now a professor at UCSF. Again, this is by now 7 years old. His age index by now is much higher, and a lot of that really is this suite of programs modeler. Here with Marc-Martin Renom, who's in fact in the CIPF in Barcelona. I showed you a picture with Cedric Notre Dame in the other lecture. He's in the same institute. And that, in fact, is sort of the reference of modeler. Modeler is a whole package. Here's the web interface. Ultimately, what it does, it has a sort of fold assignment. That means it sort of recognizes what protein in the PDB looks like your query. It does the alignment. It builds the model. It evaluates the model, asks whether the model is okay. If not, then it sort of tries to do it differently. Let me show you some steps. So improving the source, by the way, is the modeler manual. So you extract spatial constraints, meaning from copying the coordinates, that implies similar distance of all the residues, and you simply first question, all the distances that you have, are they fine? Or do you create clashing residues? So you put the C alphas too close to each other. And that ultimately, the C alpha distances, you optimize by a distribution that you see in the database. Okay? So there is no function, there is no chemical or biophysical knowledge about bond sizes. You simply model the C alpha distance from known structures. And then you fit whatever you observe into this pseudo distribution. Okay? I deliberately show the histogram here. This is not a theoretical function. It's just an empirical fit. And into this empirical fit, you simply reduce the distances. Then reducing the distances is you wobble around, and wobbling around means an optimization procedure that's shown here in terms of 7,000 iterations. And in this 7,000 iterations, in this particular case, you see there's a minimum. You can pick it, and you have one relatively optimal solution. That is not always happening that nicely, but in some cases that is. That is the part, again, that takes a lot of CPU time. The objective function from modeler is rather complicated, as you see sort of the bunch of bullet points. I don't want you to understand any of this on the level of detail. I just want to say it's not even as simple as modeler does some molecular dynamics. There's much more to it in the detail than that. And I'm going to really remain on the high level, meaning that I'm just going to pop some ideas. So there are multiple models, meaning that you're not only using one template, not only one structure. If there are more, you try to find an optimal that fits to several structures at the same time. Now, typical errors are, the side chain packing, because you have different residues, different side chains, and you don't actually know how to put the side chains and how to pack the side chains. From copying the coordinates, you only know where the C alpha sits. In which direction the side chain orients, again, there's a lot of freedom, right? The next most important problem is sort of you get the loops wrong, you misalign, we talked about that, or you completely get the wrong templates. Let's talk about the loops. Again, loop optimization or energy minimization. Ideally, when you look at the RMSD, so the difference between the three-dimensional objects that you predict and the one that is reality versus the score that you have. So ideally, you have examples such as for this protein here, where the lowest RMSD is received for your lowest score. If you have that, then you can simply take your optimization procedure and can say, in this case they call it dope score. My dope score is minimal. Dope score minimal is my chosen solution, and in fact in this particular case you do right. It's the best one. In this particular case here you see that the one with the lowest RMSD here is not the one with the lowest dope score. The lowest dope score is here, and that has a much higher than lowest RMSD at least. So you would pick the model. You could pick a better model if you knew how to. But the score that you have without knowing the structure, so if you really knew the structure, you could pick a better score. This is nonsense. If you knew the structure, you don't have to predict it. This is just to assess the method. This is just to show that not in all cases does this optimization procedure that I showed you before really find, so wobbling around, moving a little bit here, moving a little bit there, and putting all of that into energy function, not always finding the best solution. Okay? In this particular case, still the solution that you have is not that different from the best one. So the best one is 5, the one that you find 7.5 is not that different. But still is different, so you're not always finding the best solution. Now, what if the loop is missing? What do you do then? So you have, in fact, the typical case here, a rather common case where in the structure, oh, there was a P. The P was good. In the structure, you simply do not have those 2 residues. So these 2 residues are extra in your query protein. You do not have them in the 3D structure. What do you do then? I make them 3 residues. 2 is unrealistic. 2, 3, 4, 5 residues. Let's call it a loop. How would you model that? Any idea? So essentially the problem now becomes you have this point is fixed, that point is fixed, so you copy the coordinates for these 2 points, but then there are, in this particular example, 2 residues, but think about 3 or something, because 2 is a little bit too constrained. 3, 4 residues. What could you do?
Just connect the 2.
Sorry?
Just connect the endpoints of
Yes, but how? A straight line? Oh, okay. My example with 2 is really wrong. Or something like that. So I have 1, 2, 3, 4, 5. You still want the straightest line? So you don't want the straightest line anymore. Let's put it differently. So you have 5 points in between, because so what you could do is, well, let's not do what we could do. We get into that in the next step. Actually 5. I cannot count to 5. So you have 5 dots that you have to convert. What's the simplest thing you can do? So you have 5 particular residues. You don't know how to put them. Again, the straight line, I take your point, Pia. That is one solution. Fair enough. I asked for one solution, that is one. Absolutely taken. But there's a relatively simple thing that is slightly better. So the first solution would be, you take those 5, you look for the database, and you ask, are there 5 like that in loops? And what's the confirmation of those? If I find my 5 residues here, EQPIA, in the database, the 5 mirror loop, then you simply look up that loop confirmation and put it on. Now, this typically leads to more than one. Here I show you one particular loop that is used from the database. Very often you find a little bit more than one these days, and then you somehow find an average or find something that sort of approximate the best path between the several. What do you do if you don't find any? And that gets back to the next best thing you can do, which again still is not a straight line. What else could you do? So you have 5 residues, so you know the begin point, you know the end point, and you have 5 in between that you want to connect. How could you connect them? If you don't find anything in the database that is like it. Yeah? No? So, no. Anybody? Well, in principle, you could use some molecular dynamics simulation. So you could use energy minimization, and that is my concern with your straight line idea, Pia, because you will have to find something that sort of has the natural angles already, right? So, because proteins don't do straight lines. So they do sort of slightly kinky moves, and then now you could say I take the typical angles that I have from my database, but then actually you, in principle, in this idea of molecular dynamics. So I didn't want to make the next step of your idea, because the next idea is to refine it not from a straight line to a line that is most compatible with what I've seen in other proteins, and that ultimately is exactly molecular dynamics. It's not much more than that. And when you do that, here's the RMSD, here's the length of the loop. Is not the latest data, but it's 18 years ago, but it's still sort of ballpark similar. The message is, when the loops are short, you can do it, because then you have two constraints, a short loop in between. Most of the time you're right. The longer the loop gets, the more often you get wrong. Ultimately, if you get into the length of a full protein here, we know we cannot fold up proteins with molecular dynamics, and that is the problem that you're getting into here. So where exactly you put that point, how exactly these numbers are, there are essentially different ideas, but roughly loops shorter than 8-9 residues you sort of can do. Longer than that fails. That roughly is the message. There's another important method out there, or the historically second method, and those in fact are the most important arguably today, is Swiss model. As the name suggests, comes from Switzerland. In fact comes from Thorsten Schwede, who in fact did his PhD in extract crystallography. So he started as an experimentalist. And this is another person, this is outdated, this must be much more than that today. But even at the time, the age index by no means reflected the importance he had for computational biology, and simply just alone through Swiss model. Swiss model, the other person behind it, in fact, is Manuel Peitsch, Nicolas Gouet. So these are the original publications of Swiss model, and the underlying philosophy of Swiss model is the less you do, the fewer mistakes you make. So the simplest thing you can do in order to do modeling, if you want to have a completely automated method, is you align by PsyBlast because it's cheap and fast. You copy the coordinates and you call it a day. Or put differently, you return the result to the user. Because with any step of refinement, you could introduce a new error. If you just say, I copy the coordinates, and there will be clashes, there will be mistakes, because that's the way it is, you leave it to the user to refine it from there. You don't make the mistake, it's just the mistake of the process. And somehow it's a well-defined process. So Manuel Peitsch was he still is, but is a very provocative person, and he introduced this idea and simply shocked the field. Because everybody had spent all their intelligence on refining and doing things right, and he said, the less I do, the fewer mistakes I make. And he won at one of the class competitions with this thing, or came out as one of the better ones, and that sort of completely flattened the field. How can you sort of say that you don't make any mistakes and then do better than others, really, by not making mistakes? This is shocking. Today, the story is really very different. Today, Swiss model is way more complicated. There's an entire machinery underneath. So some of the slides come from Chris Wilton, but seeing the time, I will in fact cut this short, the Swiss model. The most important message really is, so there's a website, it's an automated server. The automated server returns the typical automated alignment. It returns model quality, substrains, interpro annotation, secondary structure, and then at the end, a lot of annotations. And in order to build a structure, ultimately it does sort of the same type of details of optimizing side chains or optimizing bond lengths that we saw, and that modeler has seen. The details are very different. So here the energy minimization step is very different. So many of the details are different, but the entire suite or the entire idea of this is no longer psi blast, copy coordinate by, but it's now a lot of programs that sort of come together to do this optimization. And again, most of you cannot even say that. There is not a single bottleneck here. So using different models to refine the alignment every single step on the way is where these two methods do make different decisions, and both try to make the best decision possible. After many steps, this is the type of model you get. With a lot of scores that also tell you where in particular positions the score is most likely. It's like a B value. So where in your structure is your model more correct than in other places. Completely automated, increasingly complex, and comprehensive. So on this slide here, I say there are other methods. HH-Pret goes back to HH-Blitz. What if is a whole suite to do an interactive modeling. 3D Jigsaw is something that is mostly automated. Coma is something that is very good on improving the alignment. There is a modeling suite underneath. I'm not going to go through all of these. Most people today still use one of the two that I talked about. Swiss model, because it's still the easiest. So this is the one that is easiest to be accessed automatically. It has been originally meant for people who are not experts in the field to get models, and still is better for those. Why Modeler is typically the tool of choice if you are more interactive. But the difference is really getting less and less, because Swiss model is more and more becoming an iterative tool. And in fact, by now, Modeler also has an automated, creates models for proteins out there. By the way, one tool that comes from Gert Friend, who used to be at Nijmegen, or whatever you say, is Yazara. And Yazara, I show these images here. Yazara essentially is just simply a tool that plots three-dimensional structures. And if you want nice pictures, so whatever your opinion, they look nice, and so they are ultimately coming directly from Yazara. So it's a tool that you can download. I cannot remember whether you have to pay for it. Clearly, if you're a company, so he's essentially running his own company out of it, and ultimately, so there was the front end for the modeling suite. What if? By the way, the idea behind what if was Gert had this question: what if I showed you this one residue that has a clash there? What if I have to move that residue because I see a clash? How would that affect other positions in my structure? What if I had a mutation there? What if I wanted to bind a small molecule there? What would I have to change? That was the idea, to create an interface for people who want to model questions such as this. And again, Yazara is simply the way to visualize that. Modeler, Swiss model are the most important tools, trade of the moment. And I believe there are others, ITaser here, and I will get over that. I will briefly mention this one here. What this one shows here is percentage sequence identity. So between the Q, the query, and the target. And here's RMSD. So what you see is if two proteins are very similar, and this is a bunch, it's an old method here, it's 17 years old, but for a bunch of different methods that include Swiss model at the time, which was a much simpler version than the one that is available today, and a few others. So there is sort of a spread. Some models are a little bit better than others, but ultimately they all sort of are within that window here if sequence identity is high. And then suddenly it shoots up. So if the two diverge too much, you just make too many mistakes. This is not because you don't find the right structure. This is because you make too many alignment mistakes, right? And because of essentially the type of problems that you have simply become more and more and more and more. They multiply. And that's why it's almost a phase-like transition. So you sort of stay here very close to this line for all the models. And this is in fact a result for many thousands of models, 10,000 or something like that. And then suddenly it shoots up. Now, this is one reality. So you can do the modeling fairly well if the two are similar enough. Okay? Now here's another reality that comes under the word of coverage. So at the part where you can essentially do the modeling here, say this realm here of up to 40% or something like that, what you see is typically the query and the template cover ballpark 60% of all the residues. So you're finding a protein in the PDB that has 60% of the residues that are aligned, not percentage sequence identity. So I'm asking, for what fraction of my query do I find something in the PDB? So I identify a protein in the PDB that has a similar structure at 40%. So my query and my template have 40% sequence identity. And on average, 60% of the query is aligned. So the other 40% are below 40. So 60 minus 100 minus 60 is 40. So the other 40% of the residues are so divergent that I'm not even showing them here. So they fall below the threshold, what I can identify that they have a similar alignment. And again, at this point here, roughly, this completely goes away. So even for the models, for which I still can predict the structure here, what I actually model the structure for is 20% of all my residues. Roughly here, 30% or something here, right? So even for the good cases here, where I'm not shooting up, for the few exceptional cases where I can get the right model here is in a tiny region. Okay, this is something that we have to remember. And something else that is sort of overlaid here is, where are you most likely to find hits? And unfortunately, most hits somehow, midnight zone, twilight zone, most hits are somehow left of the region where you can do comparative modeling. I'm showing a picture of René Magritte. And this picture here says, "Ceci n'est pas une pipe," which means this is not a pipe. Meaning what?
It's just a picture of a pipe.
It's a picture of a pipe, right? And that's the point here. A model is not the truth. A model must be wrong somewhere. Otherwise it wouldn't be a model. Otherwise it would be the true thing. And in this sense, this is not a pipe. The model that you build of a protein is not a pipe. In fact, Thorsten says model is a tool that helps you to interpret data. It's not something that is meant to predict 3D structure. It is something that is meant for you to understand something about that protein, that guides some other experiment, that tells you something else you should try, that brings a hypothesis up, right? The model is not the truth. And this is something, this image is something that I believe the message here is, whenever you develop a method, whatever that is, it will have mistakes. So one thing is, make it easy for users to identify where these mistakes are. Sometimes it's very instructive to not ask, okay, I get a prediction of something, model of a protein structure, where is it right? Sometimes actually it's very instructive to ask, where is it wrong? It helps you, it's a different way of zooming in. Are there things? So sometimes you may not be able to say where you're most likely correct, but maybe you're able to say, well, these positions I clearly don't believe. Well, in the most trivial way, there would be positions where I have completely different residues, a set of prolines. Or where, in fact, I look at a secondary structure prediction and I see the secondary structure prediction is different between the two proteins. So there's another extreme ways in which I could sort of suspect that the model is wrong in those cases. And there may be other ways in which you can suspect that. So my advice to you as method developer, give the tool to users so that they can find these points. My advice to you as a user, whenever you look at a model, ask yourself, where is it wrong and where is it right? Where is it more reliable? And what is it that I learned from it? And with this, I'm going to go into a secondary structure prediction. Secondary structure, and there is something wrong with my slides, and I believe it's settled in a moment. So secondary structure prediction, secondary structure is often written like that, and that brings people to assume or put the word 2D in there. And that's why often people talk about 2D, 3D, 4D, where in this rationale 4D would be the arrangement between domains. 3D would be really the 3D coordinates. Completely misleading because 2D is two-dimensional, is residue distances, and that in fact is, as I told you, contains the same information as 3D essentially, while secondary structure is a string. You assign for every single residue. The L here is in an E in an extended conformation, so in a strand. The V is in a strand. The I here is in a configuration other. And you write that as a string. It's 1D, has nothing to do with 2D. Completely misleading. So again, when I ask, and this is the slide I should have put earlier, so when I ask how much can I reach with comparative modeling now, then the answer to that is on the level of proteins here, of my 111 million, for roughly every other protein I can build a region, for a region I can build a structure, okay? That does not mean for that protein comparative modeling predicts the structure. But there's one region in that protein for which comparative modeling predicts the structure for 55 million. Okay? Again, the number that